Thank you, Dr. Srinivas, for allowing your Science Hub to visit the facility, and I appreciate you taking us around. Before we go into more details, can you briefly tell me about your background and was your PhD work?
Yes, thank you, Dr. Iyer. It is my pleasure to have you here. And of course, I think I still remember when I used to meet you in my initial days when you are heading the formulation department in Milan.
Right.
It's good to see you again. My name is Srinivas, Director Lab Projects, YMC India. And my schooling college was at Visakhapatnam and moved to this happening city, Hyderabad. Almost now 19 years settled here and worked in couple of MNCs. I started my career as a chemist and then moved to sales and technical department supporting pan-India clients, not only pharmaceutical but biopharmaceutical industries and supporting them in their business products. So I've done my PhD at Geetham University, Visakhapatnam under the guidance of Dr. Sureka. Initially I thought to do in chromatography related,
Okay.
but could not. My guide has supported in the field of plant biochemistry, done on rice research, stress conditions, but it's difficult to handle work, family, and then to do PhD at a time.
I know.
It's a tough job, but it was really a good, memorable days.
Super, very nice, very nice. What is the full form of YMC and what activities are carried out here?
Yes, YMC, it's actually our chairman name, Yamamura, a Mura company. And we obviously can say it's a one-stop solution for chromatography requirements because YMC Co Limited, it's a Japanese firm and subsidiary office is India and headquarters YMC Kyoto, Japan and manufacturing location in Komatsu. And for a global wide supply of products will be happening in Komatsu location. And YMC India under the leadership of Dr. V.K. Gupta, he is the man behind all this and we do support in different verticals, almost close to 6 verticals now. We keep on expanding since last year and every pharma company know about HPLC columns and consumables. That is our main product line and also we do manufacture equipments and DAC columns and also low pressure flash system and cartridges, and the recent additions are chiral application lab dedicatedly supporting enantiomer separations for chiral chromatography customers. And now this is a third year baby. Now where we are sitting is YMC India lab. This is a third year of operations and we do support bulk resins for those customers who want to pack their own columns. So that is again a bulk resins I'll be supporting for pan-India clients.
Super. So is this the only lab in India or you have such labs?
No, YMC India R&D lab and analytical and custom purification lab is in Hyderabad.
Super.
Only.
Super.
Now also we started in Bangalore, YMC Bangalore operations where we'll be dedicated facility for equipments and also we have a head office, YMC India head office is in Delhi, where our CEO Dr. V.K. Gupta is based at.
Very nice.
And also as I see like now we supply not only like low pressure to high pressure columns and consumable to the services and in all requirement of the chromatography needs. So I can say like proudly YMC is one-stop solution for all liquid chromatography requirements.
Super. End-to-end support.
Yes.
Fantastic. Dr. Srinivas, now we are in the biotech era. What is the contribution of chromatography in biotech according to you?
Yes, obviously, as I mentioned, pharmaceutical and biopharma industries also chromatography play a major role. We are not only like in upstream, but also the chromatography comes in downstream processing for MABs, biopharmaceuticals or biotech vaccines. So obviously, chromatography play a major role, and YMC also is in the supporting biopharma companies, not just the HPLC columns, chromatography columns, but resins. And to give an example, BryoPro, our BryoPro resin is also has been set in one of the vaccine manufacturing process in Sputnik vaccine, but unfortunately India we are not manufacturing Sputnik. But our resin has been in the process, and we supported Russian clients as well. And to be extend continuous chromatography, that is one innovative technique. I can say it is kind of breaking through the traditional technique of a single purification technique to a multi-column chromatography. So continuous chromatography is one arena where YMC is looking at, and we have a dedicated facility in USA for this technique and expanding worldwide. And this is one technique where all the three parameters of purification, like kind of purity, yield, throughput, all the three can be met without compromising on any one of the parameter. And I think Biocon Bangalore has been taking this continuous chromatography technique. They bought already our system and taking it to next level in coming soon.
Maybe you can just briefly explain to our viewers what is continuous chromatography, just for the sake of simplifying it.
To give a kind of brief input on continuous chromatography, as I mentioned, we'll be using a twin column chromatography instead of using a batch mode, a kind of a single column chromatography technique. We'll be here using a twin column and a continuous process. So maybe you can say maybe it's related to SMB. I can differentiate SMB with continuous chromatography, as SMB is a kind of, we cannot separate multiple peaks or multiple products, even though it is a high-end scalable technique. Continuous chromatography meeting all the three requirements of the client: the target purity, yield and the throughput, without compromising on any of them, and it can be scalable. And Europe, I think west side, already it has been proven in biosimilars, monoclonal antibodies manufacturing processes. Continuous chromatography has already been implemented.
Fantastic. Doctor, talking of chromatography, C18, in fact I started my career with C18 and it is still popular.
Yes.
So what is the reason that like one column chemistry can be as popular as lasting so many, almost a couple of decades or more?
I think to comment on a single line, obviously maybe you started or I started.
Correct.
C18 is the king of all gods.
Absolutely. Absolutely.
There is no alternative option of it. As we see, it still dominates, and maybe it will be remain dominant in the next decades as well. But of course, it is not the one and only solution, because in this healthcare sector coming up new products, new challenges. Obviously, like now we will be trying out, and YMC has an alternative. For example, if a product is not retained on a C18, what's next step? Absolutely. So obviously we will be looking towards what if C18 does not work. For example, a polar compound cannot be retained on a C18 column. So we have like for those kind of solutions and aqueous based columns to retain a polar compound in a reverse phase chromatography.
Like Hylic column.
Hylic column and a polar modified stationary phases, where we can retain polar compounds also in a reverse phase conditions.
Fantastic. So YMC has columns of C18 different micron size and different shapes and spherical shapes.
Yes, obviously I think hundreds of stationary phases are available, and we do have a range: reverse phase, normal phase, ion exchange, size exclusion chromatography, in all different types of, and that is the strength of YMC. Like we are not in one segment, we are in a complete, we can have a platter to a client with a complementary phases.
Doctor, HPLC is a very competitive market. So how does one decide on a good column?
That's a tricky question, obviously, because to decide on which column manufacturer or maybe which column to be going ahead, obviously which column, selection of column is again based on the technique they use. Especially in India, if you see any new model of Apple will be sold a few lakhs in just few minutes.
Absolutely.
And also maybe a low economical cost of the mobile, any new model comes in, it will be sold immediately in just few minutes. So India is such a accepting nature of they can accommodate any different manufacturers. Coming to which column to be decided, obviously nowadays the regulatory environment is keep on increasing from day to day in all the labs, not only small labs or bigger labs, everywhere the regulatory requirements. So coming to that aspect, if you have to see in that aspect, definitely we have to choose a right column manufacturer partner, not only giving the quality of the product but the presence worldwide, because once the method is developed, it has to be transferred or it has to be available for the lifetime for that product.
Yes.
YMC obviously is one of can be a better player for our we can select, because Japanese, as you see, column manufactured in Japan, even though 99.999% is not 100%. So that's why we can say we manufacture at very stringent operation conditions, and our presence is worldwide.
Super. You touched upon life cycle analysis. Now my question is, there are so many challenges any analyst faces, like co-leaking impurities and all. So what is your suggestion, how to cope up with all this? Despite good columns coming, these are the challenges which any chemist face during his life as a chemist.
And obviously, as we earlier discussed, C18 is not the one and only option. So we should be focusing on complementary phases. For example, in our lab we provide services, analytical development and validations. So every column is unique. So we will be choosing, for example, in our method development we choose minimum 6 complementary phases. For example, triad C18, C8, phenyl, PFP, helic. So EXRX, which is again a 25% carbon load column, so which is again a unique complementary phase which can be a hit for our compound. So in that way, obviously YMC has a portfolio of having a complementary phases in our basket.
Dr. Srinivas was talking about the columns. Every column has a life cycle. Yes. And they have particular life, because when you use buffers and all, column life comes down.
Right.
You have any suggestion for the chemists how to take care of the column and what is the precautions they have to take?
Right. As I think most of our pharma companies has already implemented it, because when we get a troubleshooting part, when we try to analyze the situation, if we see a 25 centimeter column, everything looks good. Maybe the first 4 mm of the column has gone something it has been clogged, or something has been a color or anything. That is what we have observed in most of the cases. So that is why everyone, I think every pharma company is now implemented a guard column before the main column, 4 mm guard column, so that we can do not want to spoil the main column and safeguard our main column, which is again a costlier column. So in that aspect, if you see protecting the main column, use a guard column. And in addition to that, as we see nowadays all the GLP labs, and we are now monitoring almost the each column life and number of injections, how many we have done. And if you see more into details, like what mobile phase we have used, everything is monitored and documented it, so that if you are trying to use it for multiple products, we have to be careful, like what product, what solvent, mobile phase we have used, and then take approximate conditions, because we see I have a client, I think I met him in the last expo, where he came down and say, like, I think triad C18, I think still it is 20 almost like close to 20,000 injections, and still it is going on. Super. And it was really appreciable. I'm not getting the repeat order but still now we have clients again, like Srinivas, maybe I am lasting, my column lasting only 2,000 to 3,000 injections. So it depends on what kind of sample nature, what trying to how we are maintaining it. At the end of the day, how my column is getting ready for the next day. So that is key part in maintenance of the column.
Good. Talking of the maintenance of the column, once in a pharma industry, one column is used for many years because it has to be filed and the same column has to be used. How do you ensure that the column consistency is maintained so that pharmaceutical industry has a reliable column at the back? There should not be any surprises.
No, obviously every column comes with a certificate of analysis. That's a basic requirement, but it's good other way around. If you see some manufacturers, maybe we cannot identify whether it is a good or bad by seeing it.
Sure.
Unless and until we perform the analysis, we get to know like whether the column is good or bad. That is why every column comes with a certificate of analysis, and the specific data will be given along with the column. So we can perform the certificate of analysis, but again, it is not the column itself. The theoretical plates define the column performance, but also there is a system role. HPLC system volumes also play an important role in calculating the efficiency of the column.
And it also depends on the chemist style, how he works.
Yes.
How he cleans his column, how he makes it.
True. At the end of the day, obviously, it is how the client maintain it and get ready for the next day.
Absolutely. And after talking of, let's say, automation now, you have analytical quality by design.
Yes.
And what is your take on that?
So analytical quality by design, obviously nowadays, as I said, we are in the health sector, and whichever technique or the technology is giving us to change our life or to adopt new things for the better human health. So obviously we will be trying to implement whatever new technologies has come up, and quality by design in an analytical way, we need to see not only just the quality of the method, but analyzing the risk assessment of the method and predetermine the quality parameters and evaluating it, and finally getting the rugged and robust analytical method is the key part. So definitely I say analytical QBD is playing a key role in most of the pharma companies.
So in future it will continue to be...
Yes.
Very nice. Okay, while taking along, I saw some big preparative HPLCs in your lab. Do you want to say something about that?
Yes, I mentioned big preparative HPLCs, obviously. In our lab, we do have from 100 ml per minute to 1 liter per minute systems.
Wow. Very nice.
And our expertise and our team expertise is the way it has been already proven for the past couple of years. We have done multi-kg projects, not just the kg projects, multi-kg projects, and supported our pan-India clients, and good that we are getting repeat orders. So it indicates that our services are good and maybe we are economical and not compromising on the quality service.
Very nice. So you have preparative right from let's say gram level up to kilogram level separation?
Yes.
Or mg level up to kilogram level?
Mg level to, so we are taking orders. I'm getting orders 10 mg, 20 mg to multi-kg.
I know.
Yes.
That is a very nice work. Okay, I also saw some students going out here. What are they doing? I mean, why so many students are here?
That is, I think, under the leadership, as I mentioned, Dr. V.K. Gupta, his aim and mission, not only we are doing business and we are running projects, but apart from that, we would like to try to accommodate or try to bridge the gap between academic and industry. Because I think I sometimes interview candidates and I see they have done a project in chromatography related. It's surprised, like every chemist has a project work done, but everything comes like I have done a theoretical, or maybe they are not having a practical exposure to really the candidates or the students. There is a gap between the industry, what industry want, and what student has. So there is some gap, and with our YMC School of Chromatography, that is one which has been established a couple of years before, and under the guidance of Dr. V.K. Gupta, we have dedicated services for that. So we have a dedicated team, dedicated system. YMC School of Chromatography, we have appointed a coordinator where she'll be going out discussing with professors, understanding there is a gap between academic and industry, and immediately we get a response, yes, that is true. Whatever our observations are, it is already not we are trying to innovate, it is practical. Yes, definitely our students has to be benefited somewhere we have to collaborate with industry, like or with industry, that is where. And nowadays I see the curriculum of even though not only the masters, bachelor's degree also they are getting in their curriculum, they have to go out and do a project work and submit a thesis, and also they have to present to the examiner kind of project what they have done. So we are trying to bridge that gap. We are providing a HPLC training, hands-on, so that they get confidence, like yes, not only whatever the professors thought, theoretical process, but like practical exposure. Maybe I see a lot of students, yes, this is the first time I'm seeing an HPLC. Maybe I don't know the make, but this is the first time. So whatever the lack of confidence to the student going out and face an interview to industry, that's where we are trying to build up the confidence of the students, giving them not only this confidence, a practical exposure, hands-on training on HPLC, and few students and working professionals. Actually, they are coming from industry also, because someone is working in a wet lab and they could not access the instrumentation lab, and someone is having access to HPLC but not to other techniques. So we do have almost close to 14 HPLC systems, LCMS, UPLC, 5 preparative systems. So we try to give them a exposure, a small scale industry exposure to them. Definitely we are sure, I think that we are already getting some positive responses from the students, like they are already facing interviews, and interviewer is really appreciating their little bit unique in their skills.
So how long does a course last, like how many weeks or something?
It is actually we have divided, it is not actually we have something a dedicated plan. Obviously we have 1 month, 2 weeks, 1 month, 3 months.
Okay.
But again we are little bit accommodate them as per their convenience. For example, right now you are seeing 60 students in our conference room. They are coming on the weekends, and almost they will be coming here only on the weekends for 3 months.
Super. End of 3 months they become industry ready, and
at least, yes, I'm getting calls from industries also. Srinivas, you train it and give it to us, so we will immediately recruit them.
Fantastic.
We can give references.
That's a compliment, yeah.
Yes,
Okay.
yes.
Good.
I think we can. The students can contact our coordinator. I think it's already in the website, as we are providing training and hands-on experience to the students. Not only just students, research scholars, and working professionals on the weekends. So we are open for any kind of support for their, which is can be important in their career life.
Fantastic. Do you also help them in placement, or do you just give them a certificate and they are?
Yes, certificate is all certified courses. We can refer the candidates, the good candidates. Obviously, every batch will be having a test, and if they are selected, and definitely we can refer them. At least we can show them the route to the industry.
So how many people have passed out? I mean,
in the last 1 year, I think almost 120 students as of now.
Fantastic.
Yes.
Good. Very nice. Good. Dr. Srinivas, what is happening in chromatography world? What's the new thing which is happening?
New thing coming to YMC India lab, as I said, this is the 3rd year of operations we completed and moved to a new bigger facility supporting analytical and custom purification services, and dedicated application lab, YMC School of Chromatography, HPLC method development and validations, LCMS method development and validations in a ISO and GMP environment. Including to this configuration, we are now adding up supercritical fluid chromatography system, which is something not new technique to India, but however we'll be having in our lab shortly SFC 750 Thar process, which is something first system in India with that flow rate, which will be in functional.
Fantastic.
Where we can accommodate projects from multi kgs in a green environment.
Lovely. Doctor, I missed asking you one question about preparative HPLC, which I recall the challenges which I faced when I was a chemist. How much to load on the column? Can you just elaborate on that?
I think this is the first basic question in every meeting, in every project. The scientists ask us like how much I can load on a preparative column, and this is something to elaborate on this. It is not. It is all based on the goal of the project. Because as we see the triangle of preparative HPLC, one end we will definitely fixing the purity of the compound what I require. So every client fixes I need 99%, 99.9%, whatever the target purity is, he will be fixing the purity of the product. And coming to the other two sides, throughput and yield. So throughput, again, if a client, again, that goal of the project, if he says I need high throughput per day, I want maybe some X quantity to be purified on a single day. So that's the throughput. If he little bit emphasis on throughput, then I will to compromise on the yield.
Right.
If maybe few projects we get, my product is very costly and I cannot compromise on my product. It is very cost. Then I have to compromise on the throughput. Per day. So how many, it all depends, again, goal of the project and goal of the client project, we take it as, and this is a basic discussion we keep on doing it. And definitely for every project we'll be doing not only development, before going to preparative we'll be doing a loading study. There we'll confirm what is the load, maximum load on a column, and maybe per day this is the what quantity you can purify. In that way, as we are manufacturers of prep systems, silica, everything is in our hand. We try to see which best phase suitable for their technique, and we'll try to see what maximum load can be done in a single day. And based on their requirement, we'll try to scale it up to kg level.
Wow, fantastic. I think, Doctor, you gave me a overview, a very nice overview of what is happening in YMC, plus you also gave me basics of chromatography. Thank you so much. Any last words for Neuroscience Hub viewers?
Yes, I think thank you Neuroscience Hub, Division of Smart Lab Tech, and Chief Editorial Team. Really, you are creating innovative, inspirative and informative videos and creating a platform, a web platform and accessibility to all chemists, and maybe you are creating a very good platform for future science technology.
Thank you so much, Dr. Srinivas, allowing Neuroscience Hub to be here and seeing your fantastic lab.
Thank you, Doctor.
Thank you.